Welcome to Module 1.4a. We're talking about neuroplasticity and tools of discovery. Here's your vocab pause if you need. So we're going to dive into biological psychology. And there are 7 major perspectives of modern psychology that we will be discussing throughout this course. When we are talking about biological influences on the brain, this is just one factor. In modern psychology, we look at all of these to understand in a more inclusive approach. So we use a biopsychosocial approach, which incorporates biological, psychological, and sociocultural levels of analysis. So for example, if we're studying depression, we're not just looking at the biological influences. We're also looking at how you've learned to deal with fears, how you've learned to or how you handle different emotional responses, how you process and think about certain events and things that happen in your life, and the sociocultural influences that are involved in how you think, feel, and behave. So we use all of those levels of analysis, but for this module and really for this unit, a lot of what we've been talking about is biological. So some of these people are behavioral neuroscientists, neuropsychologists, behavior geneticists. We talked about those in a previous mod, physiological psychologists, and biopsychologists. When we are discussing the brain, it is important to note that the brain has plasticity. This means that the brain has the ability to change. This is especially true when we are very young. Childhood, the brain has the greatest amount of plasticity, meaning that neural tissue can regrow, reorganize. Things that you don't use stop getting used, they go away. Neural pathways that we use get stronger, but also things that have been damaged can be restored. For example, if a person experiences a stroke, they may lose the ability to walk or talk, for example. The brain has plasticity, so it is possible for the brain to write again, to walk again, to speak again. If someone is blind, that region in the back of their head that is devoted to visual processing, some of that sensation to touch, like for reading Braille, for example, has been shown to be expanded into that visual cortex. So it's using a part of the brain to expand how that person is interpreting their world because they're not using the visual cortex in the way that it is originally designed to be used, right? So the brain has plasticity, its ability to adapt and reorganize depending on what's happening to it. So one way that we can study what's going on in different parts of the brain is through lesioning. This is the destruction of tissue. Now, this tissue could be destroyed because of a disease, because of trauma, because we're purposely doing it through surgery or experimentally. For example, if they lesion the amygdala, they would see a lack of fear and aggression in cats. If they stimulate that same part of the brain, they might see increased fear and aggression reaction. So that can help us understand the different parts of the brain and what they serve as a function. An EEG machine. So now let's go into the tools. So an EEG machine, electroencephalogram, is going to show us brain wave activity. This is going to be a really useful machine when we are studying like sleep disorders. If you were to get a sleep study done for sleep apnea, for example, they will give you an EEG machine, which now you can actually go and take in your own home. And then it records the brain waves, and so they can understand what's going on with your sleep and diagnose and manage. They can also diagnose and manage epilepsy through the help of an EEG machine. They can also look for brain activity in patients who are in a coma. That is an EEG machine. So it's not showing us location, it's showing us brain activity. It'll be important to note that because we want to differentiate between these machines. So the MEG, the magnetoencephalography machine, this is a newer machine, and this is measuring magnetic fields because it's getting the natural electrical activity through those through the nervous system, sending that electrical activity, that message. So in this machine that cancels out other magnetic fields, it is going to have this you're going to have this thing placed on your head, as you can see here, and then you're going to be asked to do some mental activities. So you sit there, you do those mental activities, and then they can pinpoint exactly where those activities are happening in the brain. So that is showing us specific locations, neural strength, and speed of those activities. So if they were to do surgery, they can see where exactly they don't want to do surgery because they don't want to like disrupt certain things, and where they do want to do surgery. An MRI machine, magnetic resonance imaging, is going to show us an image of soft tissue. So this is going to use magnetic fields again and radio waves, and it's going to produce this computer generated image of the soft tissue. And for our purposes today, it's going to be soft tissue of a brain. You might have had an MRI on a different part to see soft tissue. This is for our brain. So brain tumors, traumatic brain injury, any sort of just developmental anomaly, a stroke damage, dementia damage, etc. A functional MRI has that soft tissue. It's still an MRI, but it's adding in the blood flow activity. Where is blood flowing in what particular parts of the brain? And so where the brain is lit up here, for example, are showing you the general areas that this person is using in that moment. So depending on what you're asking the person to do in the fMRI machine, different parts of their brain will light up. And this gives scientists an idea and doctors an idea of where those activities are happening. And they can also use it to identify what the person might be. They can, guess 80% accuracy here of the mental activities the person was doing, reading, gambling, rhyming in this study, for example. This is just, I took this from the University of Washington's website on the Institute for Learning and Brain Sciences in case you were a little confused on the differences between the MEG and the fMRI. Both are brain activity. MEG is going to give us that precise location, and it's also, they're all complementary to each other. Like if you were doing brain surgery, you do a lot of these tools that we're going over today. So a CT scan, and you don't need to know this level of information. It's just if you were interested. The CT scan, you want to think X-ray. This is going to show us structure. See how it doesn't have the same detail as the MRI because it doesn't have all that soft tissue. So we're just looking for physical structural changes due to damage, disease, injury, and just brain development, right? That are all reasons why that might be used. The PET machine. So this is, again, brain activity, but it's not going to be blood flow like the fMRI. It's not going to be magnetic electrical activity like the MEG. It's going to be where is glucose? Where is energy being used in the brain? And where energy is being used is where activity is. So it's just another way to look at brain activity. So you can see the lack of brain activity where you might want to see it in this Alzheimer's patient compared to this brain that is typical. So last is the FNIRS machine. So this is going to, you place this cap on your head, and this machine is going to tell doctors and researchers where blood molecules are to locate brain activity. This is what it would look like. So it's showing you brain activity. Now, this machine is unique in that it is portable. This is how big it is, and you can, I'm guessing uncomfortably, but you can fit it into a backpack and hike it to a location that is very remote where people do not go to hospitals that have these other machines. And so we are able to look at the brain scans of other populations, so to include them in the human genome research where we might not otherwise be able to. So that sums up Mod 1.4a. We remember biopsychosocial and how we don't just use biology. Lesion destroyed tissue, CT X-ray, EEG, brain waves, think sleep studies. MEG uses that magnetic field for specific neural brain activity. fMRI, blood flow for brain activity. It's a little more general than the MEG. MRI, soft tissue. PET, brain activity again, but it's looking at glucose and where glucose is being used. And the FNIRS is that portable machine that is showing you blood molecules and where they're active in the brain. All right, I will see you guys in class.